Hello, and welcome to Basic Rigor for Experimental Design. This is a professional development core from Transcend's short video series. I'm Mark Williamson. Let's get started. So, the goals of any experiment can roughly be thrown down as generating robust and unbiased results in order to search and find truth about the natural world, at the same time accepting the imperfection and incompleteness of science. Science is never finished. There's always openness to more data, more conclusions, and you can never run the perfect experiment. But you can certainly run a good, robust, rigorous experiment. And so that's why we'll talk about experimental design today. So, what is experimental design? Well, a short definition would be a detailed plan for collecting and using data to identify causal relationships to have a reasonable chance of detecting effects and testing hypotheses that reasonably answer your research question. So, getting an experiment set up so you can actually get results of what you're interested in with that experimental question. So, a good experimental design really has a lot of good downstream effects. So, how you set up your experiment will define the methodology, how you go about actually collecting data, etc. It will affect your analysis, how you can actually analyze your data, what sort of tests you can use, what sort of results you can pull from that. It'll color your interpretation of the results, whether they can be trusted or not. And it'll affect your results, how you report them, and whether you can even publish them if it's high enough quality, if you've considered everything that needs to go into a well defensible experiment. So that's what we're going to cover here in 5-ish rules of rigor to have a more rigorous and more defensible experimental design. We'll talk about question clarity, having a complete variable set, making sure you have sufficient samples, variation and error control, and finally, an appropriate analysis plan. So, first part, question clarity. You really want to clarify what question your experiment is trying to answer. That is, you need to have a good hypothesis. So you really want to define your hypothesis ahead of time. Again, a hypothesis is the proposed testable answer to a research question. What you don't want to do is sort of just muck around with data and find something and then sort of backfill the hypothesis in from the end. That's not particularly good science. So you should really have clarity before you even analyze your data, before you even collect your data, of what you're trying to answer. What's the question and what you think the proposed answer is. Next, you want to ensure both testability and falsifiability. That is, you need to be able to test your hypothesis and not only be able to potentially prove it. Really, it's about making sure it's falsifiable so that if you fail to disprove it, maybe you have something there. So if your experiment doesn't lend itself to falsifiability of your hypothesis, it's probably not good science, it's probably not rigorous science. You need to make sure you have that built in. And then sort of the third subset in this question or clarity point is you want to be able to differentiate between planned and exploratory analyses. So there's nothing wrong with, as you're analyzing data, coming up with more questions or coming with interesting things to look at that weren't part of your original hypothesis. Because once you actually have the data, you might not have been even able to conceive that exploration without actually having the data in hand. However, you want to make clear, in reporting results or talking about your results, that you are saying like, yeah, this part here that we didn't specifically plan for, it's exploratory. So there's caveats with it. It needs to be followed up more rigorously as opposed to your planned study, where you've defined your hypothesis ahead of time, you made sure it's testable and falsifiable. That's more defensible in the moment. Next up, variable set. First, you want to highlight primary predictor variables. So typically you have an outcome variable. You could call that the Y variable. You could call that the response variable, and then you're typically studying things that you think predict that outcome. So if it's death or incidence or even the number of birds nests, what variables go into it? Is it going to be location? Is it going to be, treatment group or whatever? You want to really have clear in your mind what the important variables are that you're going to use to try to predict or explain your outcome. On the other side of these things, you want to ensure you have all relevant, what I would call confounding variables included. So confounding variables are usually ones you're not the primary predictor variables. You don't necessarily care about them, but you know they might affect them. So maybe they're a known risk factor for a disease where you can add another risk factor, a novel risk factor, or treatment for a disease. You want to still be able to include those confounding variables to control for that. And it's really important to think about this ahead of time because if you go and do a study, do an experiment, and you forget to measure variables or collect those variables, you might not be able to get them after the fact. And then the third thing is have clear rules for variable exclusion. Sometimes a variable you might need to exclude. You don't want to just do it willy-nilly, like just ad hoc, just because it doesn't make the data nice or doesn't follow along with your proposed hypothesis. You want to have a clear, justifiable rule for like maybe excluding a variable. Maybe there's too few entries or the groups can't be collapsed or something like that. But having those ahead of time so it's justifiable and not just because. All right, moving on to sample size. You want to make sure you have enough samples of whatever you're measuring, whether it's a clinical trial with patients, whether it's sites and you're examining trees or something, again, on the ecological context, or you're doing a animal study. You want to make sure you have enough samples. And so I would say the first thing to do is make power analysis the default. So power analysis can be a little bit tricky, especially over more complicated design, but it's meant to elucidate how many samples we need to reasonably detect an effect, based on background. And so while sometimes it can be a pain to do or maybe feels irrelevant, really putting in the work then to make sure you have enough samples ahead of time is going to pay a lot of dividends. You don't want to run a study and then realize it's way underpowered and then you've just wasted all that time, effort, and money on a study that wasn't even going to be able to show your effect anyways with that number of samples. As a complementary approach, you should also check the literature for sample number and comparable studies. So see what other people have done in the similar experiments and making sure kind of how you're designing your experiment, how you're proposing it, kind of tracks at least with other people. That doesn't mean you need to always need to do as many samples, or maybe even need to do more and make it more rigorous, more robust. But it's good to have your have a pulse on what other people have done in the field and what sort of is considered an appropriate sample size, even above and beyond power analysis. And finally, you might consider dataset simulation because sometimes it's hard to really get your mind around how many samples you need, how you need to set things up without playing around with data itself. And now you probably don't want to have to do all that playing and figuring out when you've collected data and then it's too late to make any changes. So thinking about simulating your data, having a similar simulated data set of what you think you're going to collect and be able to play around with it and look at effect size, look at outcomes, that can really be helpful if you have the knack of doing that in sort of a statistical software like R or SAS, etc. All right. Fourth here, we have error control. You want to think about metadata and random effects when you're designing what sort of information you're collecting. So metadata would be, not necessarily data that you're necessarily going to use, but maybe it ends up being important. Like you're sampling outdoors, maybe season, maybe month, maybe environmental conditions, temperature, that weren't initially part of your study, sort of just metadata, but maybe that's important and it can help you control for variability or track down errors or confusing things about your data. And then kind of on the more deliberate variables that you don't necessarily care about or data that you don't necessarily care about, but you want control for is that those random effects. So those are a little bit different from confounding variables. Confounding variables are variables that you typically think maybe have a role in outcomes. Say you're looking at a health data set, you're looking at treatments, but you also include something like obesity or age in your model because you know those are risk factors for disease or mortality. Whereas a random effect might be something you don't actually care about them at all. You don't think they have any, I mean, real effect, but they something like the clinical center they came from, or some sort of blocking design might be a random effect. You might want to consider that. Again, this goes into more complicated experimental design. So if your design is going to be more complicated, make sure you think of that ahead of time. All right. Another thing for error control is check over data entry procedures for manual errors. So obviously, there might be sort of error in the sense of like variability in your data set just sort of built in, the sort of statistical error. But there also might be like real errors in the sense of like numbers that aren't supposed to be what they were. And so it's important to maybe look through all the steps where like data is collected and make sure that there aren't any obvious errors. You know, if you have a spreadsheet and you have weight, and then like there's one weight that's like 200,000 pounds, like that's probably a data entry error, and you need to fix that. Otherwise, that's totally gonna throw off your results. So, you know, while we think, oh yeah, that shouldn't happen, like these manual errors, but they certainly do, and so you want to make sure you're checking for them so they don't slip through your grasp. And finally, you want to include uncertainty in your statistical summaries because, again, no matter how much you control for things like random effects, no how much you make sure you don't have manual errors, there's still going to be uncertainty. There's still going to be messiness, inherent messiness in the data. So you want to, you know, in addition to say like means, you want to have some sort of measure of uncertainty, like so means and standard deviations. Maybe you have hazard ratios, and then you're going to want 95% confidence intervals, etc. Always including uncertainties in your statistical summaries really shows you not just like the estimate, but how certain you are of those estimates. All right, fifth and final, we have the analysis plan. You want to make sure you list and test statistical model assumptions because you know a lot of models, and in fact all models have certain assumptions. So if you're going to run a linear regression model, it has assumptions like normality of data. If you're going to run something like ANOVA, there's going to be homogeneity of variance included along with normality, etc. I mean, there's more. I'm just giving examples. And if you don't meet those assumptions, your analysis isn't valid. And so you might have to do a non-parametric test. So it's good to think of ahead of time and list in all your model assumptions and then actually go through and test them to make sure they hold, that your analysis are telling you what reliably telling you what you think they're telling you. You should also consider a generalized linear mixed model as the default. So a generalized linear mixed model is a very flexible model. And essentially, it can do everything like a t-test, ANOVA, linear regression, logistic regression, other kinds of regression, sort of all in one. And you can tune the parameters and sort of fit a lot of different data sets while still meeting assumptions. And so if you don't know anything about generalized linear mixed model, but you know basic stats, it might be a good thing to learn about and really pay dividends to your analyses. And final, with analysis plan, you want to make sure you have multiple checks on model interpretation. You know, say you run an ANOVA model, you want to look at the output, probably want to do a post hoc test. You probably also want to run, you know, make graphs, things like that, or make sure you're checking, you know, the model with another similar test or things like that to make sure you're interpreting the model correctly and you're not getting confused, you're not getting fooled. Especially big complicated models which can easily get away from you. You want to make sure you clearly understand what it's telling you and you're not sort of tripping up at the finish line when it comes to model interpretation. All right, with that, hopefully this was helpful to give you ways to improve your experimental design rigor. We have trust and clarity, making sure you have a complete variable set, sufficient samples, variation and error control, and having an appropriate analysis plan. Thanks for joining me, Mark Williamson. I will see you next time.